Yes, the name of the substrates are four. That is propionyl-CoA, glycerol, lactate, and amino acid. That is glucogenic amino acids, alanine, and also glutamine. Alanine in liver, glutamine in kidney. So now we are going to discuss about how these four substrates will give glucose for the fasting state or in the starvation state to maintain the blood glucose, right? The first one is glycerol. Actually, it is a waste product from lipid metabolism. After lipolysis only you will get glycerol. After hydrolysis of TAG, that is triacyl glycerol, after hydrolysis only will release glycerol, right? This glycerol is converted into glycerol 3-phosphate with the help of glycerol kinase. This is a very, very important MCQ point because glycerol kinase is not present in adipose tissue. This is present in liver and kidney only. This glycerol 3-phosphate again converted into dihydroxyacetone phosphate. We already discussed in the metabolism glycolysis. Usually glycerol 3-phosphate is converted into dihydroxyacetone phosphate, which enters into the pathway of gluconeogenesis and then produces glucose finally. So this is how the glycerol is synthesizing glucose in gluconeogenesis. The two organs are liver and kidney only, but in adipose tissue there is no glycerol kinase, so no place for gluconeogenesis. Second one is lactate. You all know the end product of glycolysis is pyruvate in aerobic conditions. In anaerobic only the lactate is produced. This lactate is actually in the situation of strenuous exercise or vigorous exercise in the muscle only they will go for anaerobic glycolysis and produces lactate, right? In muscle this glucose produces pyruvate. Pyruvate is converted into lactate with the help of lactate dehydrogenase enzyme, right? This lactate enters into the liver through the bloodstream, that is in between blood. Through the bloodstream they enter into the liver. In the liver the lactate is converted into pyruvate. After that this pyruvate only enters into gluconeogenesis, finally produces glucose. You all know that TCA cycle final end product is oxaloacetate. That oxaloacetate is synthesized from pyruvate. This oxaloacetate is present in mitochondria, which is shifted to the cytosol and produces phosphoenol pyruvate. These are all the glycolysis steps. But you should know the lactate is converted into pyruvate. This pyruvate again enters into gluconeogenesis pathway, produces finally glucose. This glucose is again transferred to muscle for their energy. This is the Cori cycle, that is C-O-R-I, Cori. It is nothing but transfer of lactate from muscle to liver, known as Cori cycle. Okay, how the pyruvate is converted into lactate and finally how the lactate is converted into pyruvate finally produces glucose is the concept of Cori cycle. Third one is alanine. There are so many glucogenic amino acids are there. Usually alanine will come for the gluconeogenic pathway in the fasting or starvation situations. When you will get alanine? Breakdown of protein and dietary protein. This alanine synthesized in muscle. The reaction name is transamination. See, in the muscle, glucose converted into pyruvate. Pyruvate is again converted into alanine. How? Alpha amino acid going for transamination reaction finally produces alpha keto acid and alanine. This is one transamination reaction occurs in muscle that produces alanine. This alanine enters into the liver. Why? Again they will go for transamination reaction, that is alpha keto acid combined with alanine produces pyruvate and glutamate. This pyruvate again enters into the gluconeogenesis pathway, finally produces glucose. So our concept is with the available non-carbohydrate substances, try to bring out in the gluconeogenesis pathway, finally produces glucose. Okay. So in the liver, this glucogenic amino acid alanine enters into the transamination reaction, converted into glutamate and pyruvate. Pyruvate enters into gluconeogenesis pathway and finally produces glucose. This is the third non-carbohydrate substrate. The last one is propionyl-CoA. This is best example for hard chain fatty acid. Propionyl-CoA is, after oxidation of fatty acids, you will get propionyl-CoA. This propionyl-CoA converted into D-methyl-malonyl-CoA and L-methyl-malonyl-CoA. This reaction is very, very important because the enzyme name is also important. The coenzyme name is also important. So we need three molecules, one molecule of CO2, one molecule of water, one molecule of ATP for the conversion of propionyl-CoA to L-methyl-malonyl-CoA. The enzyme name is propionyl-CoA carboxylase biotin, that is carboxylation reaction vitamin. Biotin is the coenzyme for this reaction. This methyl-malonyl-CoA finally converted into succinyl-CoA with the help of methyl-malonyl isomerase. This is the enzyme name. For this isomerase, we require vitamin B12, right? Then only the reaction is converted and produces succinyl-CoA. You all know about the fate of succinyl-CoA. Enters into TCA cycle, and this TCA cycle, that is oxaloacetate, up to oxaloacetate the TCA cycle will occur. Then this oxaloacetate will be converted into phosphoenol pyruvate and finally produces some amount of glucose through gluconeogenesis. So this is how propionyl-CoA enters into TCA cycle and finally produces gluconeogenic pathway glucose. Okay, so these are all the four substrates for, that is non-carbohydrate substrates for gluconeogenesis.